guys, welcome back to Jeevashastra Abhyasa. I am your educator Niranjan Hegde. In this video, we are going to continue the topic which has been left, that is principles of inheritance and variation. So in that topic, we have completed the monohybrid cross, the test cross, the back cross, and some other topics. We have cleared it. So now we are going to continue the topic with, if you haven't seen that whole 7 videos are there, so just I'll put the playlist in this YouTube iCard, so just go and watch it. And today we are going to discuss about after monohybrid cross, so what Mendel had did, and that is by the results what he have got with monohybrid cross, we are going to discuss about the dihybrid cross or inheritance of two genes. Let's do not waste the time. Let's move to the topic. The inheritance of two genes or dihybrid cross. So here Mendel used, first of all, Mendel used two different characters, two different set of characters, we can say set of characters or alleles. Okay, and he did the experiment in the same way as the monohybrid cross. Okay, he did the same in the same way, and he took two different set of characters. As I told you, the characters he took were the first one is shape of the P, as you all know that this Mendel did all his crossing and genetics experiments over the pea plants, right? Okay, and the second one he took the character is the color of the cotyledon, and the shape of the pea, where there are two types of shape were there. One was wrinkled, and another one is round. Okay, one is wrinkled, another one is round. So there were two colors of the cotyledon were there. One is green, and another one is yellow. Okay, and he named the color as the green, the yellow as capital Y. Okay, remember this. He named the color. Does not exactly what the alleles represents. He named it for his easiness, or to do the maths or the statistics. For that reason, he did this one. Okay, he named it yellow Y capital, and this was the dominant allele. Okay, dominant allele. Then comes the round. Capital R. Okay, these were the two dominant colors. That's why I have written in the red color and for differentiation. Then comes the color green, and this as in the monohybrid cross also. So it's been a recessive character, which suggests that he named it as small y. And the next one is the shape of the P, and the recessive type here, that is wrinkled, that is small r. And this were the characters he took, and he used them together in a combination. He took the two dominant one together and the recessive one together. So that made not a lot of difference was made, but let's see, the results were very shocking, and yes, of course. So this paved the way for the change in the genetics. Okay, so and let's learn how we did it using the statistical way, and by the presenting it in a flow chart. Okay, firstly he took the parent generation parent, I'll write P for parent generation as pure homozygous. Remember it, homozygous. I'll not write homozygous here. Homozygous round yellow. Homozygous, remember that, homozygous round yellow, which is being crossed with what? And the cross symbol is this one. Okay, crossed with homozygous wrinkled green. Okay, this is homozygous round yellow, and this is homozygous wrinkled green. Okay, so two different set of alleles have taken, two different characters are taken, and now in a single plant, and now it is being crossed, and after crossing, not after crossing, before crossing, it is that there should be a generation of gametes has to take place, right? And the gametes which came out, were gametes which came out were after the process of meiosis. Remember that it is after the process of meiosis. The gametes have came out, and the gametes were capital R, capital Y. So I'll let you know at the end of this topic why it is capital R and capital Y. Here small r and small y. Okay, so these two came out as a gamete and they fused itself, right? They fused and they have came out with the F1 generation. Remember that F1 means first filial generation. 1 marks question it is, first filial generation. Okay, first filial generation. And here it comes to be, so when it mixed capital R, capital Y, small y. You may ask me why I have written like this. I might have written capital R, capital Y, small r, small y. So with respect to the dominant and the recessive characters, I have written like this. Okay, and we got a homozygous or heterozygous. So it is both the dominant characters have expressed it itself. You can see capital R and capital Y, which has given us the round yellow again, right, as that of the dominant character. But here, so is it homozygous or heterozygous? No, it is a heterozygous because of the combination of two different kinds of alleles, right? Okay, so then it is heterozygous round yellow. Okay, yes, so we got F1 generation as heterozygous round yellow. Did he stop here even? No, he again moved to the he wanted to check what is the result when he is going to self. If I self, what I am going to get? That the Mendel thought, and he selfed this F1 generation with itself, and he was shocked to see very different angles of characters expressed in the F2 generation, which we are going to see in the here. Okay, so here we got to see. So he selfed it. So I'll show you now. So after the F1 generation, what he got, that was heterozygous round yellow. He crossed themselves, or he selfed it, or he inbred them. So inbred, how inbred them? This is the parent. He took the F1 generation as the parent, and the inbred was capital R, capital Y, small y. Inbred, I'll write here, inbreed or selfed to with what? The same one, capital R, capital Y, small y. That is heterozygous round yellow. Heterozygous round yellow. Both are the same one. I'm not going to write over there again. And the gametes came out where very lot of gametes came out. How many can you count here? Yes, the combination is of 4 different varieties of gametes came out. And that gamete is I'll show you one by one. Okay, you just write down one by one. Capital R, capital Y, the first one. Capital R, small y, the second one. Okay, next is small r, capital Y, and the last one is this small r, small y. So he got 4 different types of gametes from one parent, and the same combination of the gametes he got from the second one even. So I'm not going to again write that also. So you have to write during the examinations, you have to write both the sides. You have to fill up with the heterozygous round yellow and also with the gametes. So there are how many gametes are there now? So I'll write, of course. Let's write it. Yeah, 4 gametes I have written. And if we do the simple crossing method, so it will be a very hectic confusion. There will be a lot of confusion which is going to happen. So what we have to do, so we can put it in a Punnett square, which we have discussed in the monohybrid cross itself, which I'm going to put in I2, so you can go and check out that particular thing. So Punnett square, we are going to do it. So it will take a lot of space. So I'll just put it out, I'll rub this and I'll come with the Punnett square. Yeah, so we got it, and this is what the Punnett square looks like, where we are going to arrange all the gametes on the top and on the side of the table, and at the one corner there will be we are going to consider one the top part, either the top part you can consider male or female partner, and also the this one you can consider male or female, anyone you can consider male and female. When we cross it, we are going to get 16 different charactered plants. Remember that, not 16 plants we are going to get. We are going to get 16 different charactered plants we are going to get. So you can see what are the different characters of plants we are going to get. The 16 different charactered plants are there, but if we phenotypically see it, phenotypically means if we physically see it, we can only look into the 4 different charactered individuals. 4 different charactered individuals only we can see. So that is, which are those? That is round yellow. You can see also round yellow, and round, this one is round green. This one is round green. You can see, if this is round yellow, where there is a the bigger Y and the bigger R is that is round yellow. All of them are. And where there is bigger Y, bigger R and a small Y, it is known to be round green. Okay. So then it comes to the point where we can see the next 2 characters are over, and the last you can see the only one single character individual, that is wrinkled green, right? I'm going to write WG as wrinkled green. And another one character is there. That is this one. That is wrinkled yellow. This one, even this one also, wrinkled yellow. I'm going to write WY. So phenotypically, we are going to see only the 4 different character individuals, which gives us the ratio, the phenotypic ratio. I'm going to write over here, phenotypic ratio of, I'm going to write in different ink because it's very easy to identify, 9:3:3:1. So this is the phenotypic ratio what we get. 9 round yellow, 3 wrinkled yellow, 3 round green, and 1 is wrinkled green. So how you can identify? Easy. Capital R, capital Y, if comes, it is round yellow. If capital R small y comes, it is round green. And if it comes to be small r capital Y, that is wrinkled yellow. And the last one, that is wrinkled green. You can also put up for identifying 9 different kinds of things. You can put up this. There will be particularly, you can see see, this is the triangle you can make. 1, 2, 3, 4, 5, 6, 7, 8, 9. Total on the line you can see the 9 different, and 3 and all other you can easily identify. But when it comes to the genotypical ratio, it is 16 different individual plants, right? But 16 different characters you can see. In that also very small amount of characters just diversify itself. So I'll write the genotypic character ratio. The ratio stands to be 1:2, 2:2, 2:4:1, 1:2, 2:2, 2:4:1, 1:2, 2:2, 1:2, 1:2, 1:1. So that means how you can identify this genotypic character? Easy. Variation of the alleles. Ultimately you can find out. So if you sit down and you can find out it. So I'll show you one or two for you. The first one and the last one you can see easily. One round yellow, homozygous round yellow, homozygous wrinkled green. The two you can I'll make you identify. This small y has how many times it is repeated? Just check it. The capital Y and the small y with capital R and capital R. How many times it is repeated? Only twice. This one. These two you can see, right? So these two twice it has been repeated. So like this, there are other different varieties has been repeated. If you check, if you just sit quietly and check it, you can easily identify it. So this is what the dihybrid cross or inheritance of 2 genes, and what he got. These two are very important. If you write this itself, only you will get the marks. So what is about the marks? So it comes for, basically it comes, I'll rub it. So you can take, before this, before rubbing it, you can take the screenshot of it. So what about marks? It comes for 5 marks in the board examination. Repeatedly has been asked for several times it has been asked. The total, whatever the number is there, that would be 5 marks. 5 marks question, the inheritance of 2 genes, and 1 marks question also may come. So on this result, he deduced, or he has given us a very important law of in one of the very important law of inheritance. So what is that law? Let's see. Yes, on the basis of the dihybrid cross or the inheritance of 2 genes, he deduced a very important law of independent law of genetics, that is law of independent assortment. Law of independent assortment. The name itself suggests what it is. Simple, very simple. If a factor is having 2 or more character, if a factor is having factor with 2 or more characters, if there is, it is there, the characters are going to separate. The characters are going to separate. The characters are going to separate. When the characters are going to separate, when there is a formation of and this character is going to separate. That is a very important law thing comes, that is independently. Independently, it is going to assert itself. It is going to not only separate, it is going to separate and also assert themselves with different character individuals during the formation of assert and assert during the formation of gametes. Assert during the formation of gametes. So this is the law which changed the whole scenario of genetics, and this law is now also accepted all over the world. And this law came from the experiment inheritance of two genes. So this is what the inheritance of two genes stands for, and 5 marks. You have to complete the answer with this particular law of independent assortment, and then you're going to get the full marks. So this is up for this video, and just go and like this video, share this video with your friends, and let's grow together. Jai Hind.